Hi everyone, and welcome to today's Biology Daily Booster. This is number 17 in our Paper 2 series, and it is advanced information listed for both the Foundation Biology and the Higher Biology candidates. What we're going to start off with then are monoclonal antibodies. Now, a monoclonal antibody is produced in the lab using a special type of cell called a hybridoma. A hybridoma is a cell that we've made by fusing myelomas, cancer cells, and lymphocytes, a type of white blood cell that makes antibodies. So the hybridoma is the cell we've made by joining the myelomas and the lymphocytes. We call these things monoclonal antibodies because they're produced by this single clone of cells. So once we've made this hybridoma, they are all going to make the same monoclonal antibody. So that means the monoclonal antibody we make with that one particular clone of cells is going to target one specific type of cell. The way they do that, they bind to the antigens on the surface of our target cell, kill it or prevent it from operating effectively, depending on obviously the mode of action there. They have asked for a bit more detail in the past about how we make these monoclonal antibodies. And these are kind of like the key points that were on that mark scheme. So we've got genetically modified mice being injected with our required antigen first of all. So GM mice injected with required antigen. They produce antibodies to that specific antigen. We collect the lymphocytes and we fuse them with myeloma cells. That makes a hybridoma, and those hybridomas make the monoclonal antibodies. So those are kind of like your key points, if you like, to make sure you always include in the answer. The reason that we have to use this fusion with myelomas is lymphocytes can't actually survive outside the body. And myelomas are pretty special because they can reproduce indefinitely. So once we've made it, we can just keep making more and more copies of that hybridoma and therefore make large quantities of these monoclonal antibodies. In terms of what we use them for, pregnancy testing is one of the earliest uses. So what we have is HCG, so human chorionic gonadotrophin, and that's produced from about 2 weeks after conception. So before you kind of really show anything, you are making this particular chemical HCG. If we've got monoclonal antibodies that bind to HCG, we can use them in a pregnancy test in a colour change reaction. So basically what we have on that little pregnancy test stick is a little band of monoclonal antibodies that are actually going to be bound onto particular colour dyed particles. So that as the urine containing the HCG comes into contact with those monoclonal antibodies, it's going to move that little coloured particle up the stick, and then it sticks on a little line on the stick itself. So you get that little positive test if the HCG is present. If not, doesn't drag the little dyes up, doesn't do anything instead. Second one, detecting disease. This one is something you should be quite familiar with from COVID tests. Then again, we can use them to bind to specific antigens to confirm whether it's present or not. We can use monoclonal antibodies in treating cancer because we can use them to target specific cells to then kill off those cells, or at least prevent them operating as they should normally. We can also attach drugs or radioactive substances to them, so again we'll deliver that to those target cells. Whole purpose of doing that, it minimises damage to any surrounding tissue. Always good when talking about any cancer treatments because the side effects can be quite severe. And what we've actually done so far is we've developed these monoclonal antibody treatments for some cancers. So breast cancer, stomach cancer, bowel cancer, we do have these monoclonal antibody treatments for those, which could be used in the right situation. And we are carrying out research on other types of cancer all the time to try to improve this. If there are any bits from today's Daily Booster you want to look at in more detail, then obviously we've got the video on the main channel. There's a section in your revision guide on it. I hope there's sections in your classwork on it. And of course, have a look at some of those past exam questions on the OCR website. do look at those papers before you go into the exam. Don't make the first one you see the one of the exam itself. And don't forget to join us again tomorrow for our next Daily Booster.